why don't we start with a little bit of history? Because I believe that in order to understand the present and the future, you have to look back a little bit. So how did we get here with our understanding of bioelectricity, going all the way back to Galvani and Volta? What can you tell us about those two gentlemen?
So essentially, they both were working at a time when all of science was still getting to grips with what electricity even was. At this point, around 1780s, 90s, people were still sort of rubbing, glass tubes with silk cloths and marveling at the fact that then everything would become quite staticky and, doing these sort of parlor experiments with, this one with the levitating boy who would be tied up in silk and then, activated with static electricity. And then you would see, little bits of fluff and little bits of lint come sort of attracted to him as if he were magnetic. And so people were still, I guess by the 1780s and 90s, people had gotten a bit wiser about it, but it wasn't that far in the past that it was a huge mystery what any of this could possibly mean. And I think putting yourself in the shoes of people who were absolutely confounded and had none of our, sort of contemporary understanding of how, the rules work and the laws, kind of gives you a good insight into what happened when Luigi Galvani, who was an anatomist from Bologna, first started to consider that maybe this substance that they were still, sort of using as an experimental approach to zapping frogs and zapping all these animals and seeing what happened to them and which particular way they kicked their legs. Why he started to think maybe electricity wasn't just contained in materials like amber or Leyden jars or lightning. He started to think maybe there was a unifying principle by which the juice that took our intention and propelled it into action, that was its own form of electricity. And so he really felt almost, I think, called by God. He was very religious to bring this understanding into the scientific realm. And he was just a very different creature than Alessandro Volta, who was very much sort of what we would consider an early physicist, like an experimental physicist. At the time also, what makes it sort of difficult to stand in their shoes is that science hadn't yet bifurcated, or not bifurcated, like infinity-furcated into all the different disciplines that we now understand, neuroscientists, biologists. People were still calling themselves natural philosophers. So you could study this massive panoply of things across different silos and put together knowledge from these different things to generate greater insight, which I think is something that's been, that's something that we've traded in for much greater understanding of specifics within scientific silos, but of getting ahead of myself. So Volta was very much of the experimental physicist mindset. His burgeoning area of science, they called themselves electricians, which had a very different connotation back then.
Oh, that's interesting.
Yeah, so that was something very, it was sort of the 1700s equivalent of calling yourself a rocket scientist. So he was really ambitious. He had, he wanted to make a name for himself. And when he first saw Galvani's manuscript, he was incredibly excited by it. And then within a few months, he had changed his mind because he had an alternate theory of why frogs, sort of twitched their legs when you touched them with the proceeds of a Leyden jar, which was at the time like a big capacitor that people used to store electricity so that they could use it for experimental reasons.
Right.
And they had, this goes into copious detail in the book, but they had basically this just absolute war that at one point enlisted basically every scientist in Europe. And when you read like the way that the way that they talked to and about each other, it feels very contemporary. They're really like going after each other.
Yeah, I wonder what it would have been like if they had something like X.
Oh God.
Imagine.
Well, yeah, slightly less florid. Maybe no, probably not less florid, to be honest. But yeah, it did feel like a Twitter war when I was reading the stuff, the letters. I thought, well, God, things, don't change. People get all hopped up on their outrage and then they, choose sides, and then sometimes they flip sides when it seems more politically exigent. Anyway, so the reason that I wrote about this fight in so much detail in the book is that when I was looking into initially this, the initial sort of bifurcation of electricity and biology, which I think most of us can agree are just these wildly separate tracks in science now. One of them is this sort of, engineering thing that's for power lines, and the other one is not related to electricity.
And you're sorry to interrupt, but you're often, as a child, taught as a biological creature, don't mess with electricity. You're gonna hurt yourself, right?
Yeah, because it's almost like it's the opposite of biology. Almost
exactly.
Do not cross the streams. Anyway, but when I was looking into why that was, like, why do we treat them so differently when things seem like they aren't so separate? That's what I came to, this early just schism between Galvani saying electricity is relevant in biology and Volta saying you're absolutely wrong. And I didn't get into this too much in the book because it does, like, there's a big philosophical aspect of this concerning, and religious, considering vitalism, which was this idea that, life is special, it's imbued with the breath of God. And, so people like Volta were like desperately trying to separate themselves from this. They were saying, this is physics, this is engineering, this is something we can use, this is practical. Let's get all the woo out of it, right? So they were just scraping everything. And so people like Galvani got caught up in this unfairly. And that was sort of the thing that I got really obsessed with writing the book was, there are a lot of examples of BS in bioelectricity. I mean,
sure,
you can't throw a stone without hitting one, but there's legitimate research that gets thrown out with the bathwater all the time. So I guess the project for my book was get the good stuff, separate it out of the garbage, and then, put it all in one place. Do you know what I mean?
Sort of the state of the art of where we actually stand with this sort of thing.
Exactly.
Yeah. And what I find really interesting, it occurred to me, because you brought up the idea of vitalism and sort of the idea of the spark of life. And what we see oftentimes when you start pushing the envelope of science, it ends up in science fiction. And what do we see in the early 1800s is Frankenstein. Mm-hmm. And you have electricity coming into effect and animating a corpse, right? So that occurred to me, and it's interesting how quickly it made it into common, popular literature at the time. Yeah, but sorry to interrupt. And one more thing, I think Volta is credited for inventing the battery. There are other people who make that claim, but just so people are aware, I mean, he's a big part of what we know in regards to industrial or technical electricity.
Absolutely. And one of the interesting aspects of that is that it was because of his fight with Galvani that he had the idea for the battery, because it was both of them looking at electric eels and their electrocytes that gave Volta the idea that if you stacked differently electric materials and separated them with salt water, then you would get a flow of electricity, which you had never gotten before. This is hard to overstate how consequential the battery was. I mean, I think, everybody knows that, but I mean, at the time, it opened up this, nobody had ever had a tool that dispensed a sort of steady amount of electricity for scientific research. Before that, it was like, these like clumsy Leyden jars that basically just like zapped the heck out of you when you opened it. There was one guy, he tried to cram his Leyden jar so full of electricity that I think he ended up in the hospital, and one of the guys he worked with died. Anyway, so it was quite unpredictable work with electricity. And then you get this battery, and then in no time at all, now you've got people separating elements, discovering elements, just being able to do really good scientific research. And then you get the telegraph wires, then you get electricity, and it just takes off from there.
Now that schism that you mentioned, that battle in regards to bioelectricity, it lasted after their death, right? It wasn't until essentially, from what I understand, the 70s when we have the invention of the patch clamp that really sort of put a final nail in the coffin, right? Tell us a little bit about that.
Well, so Galvani was sort of the only torchbearer for his idea. He was really he was grabbing from really this huge sort of all-you-can-eat buffet of different disciplines, anatomy and electricity, etc., bone density studies in birds. But he just had this real polymathic approach. And there was nobody really left to carry his torch after he died. That's also a really tragic story I get into in the book. Just like his ideas were rejected. He died penniless and brokenhearted. And so Volta, then his career takes off. There is one guy who tried to carry the torch for Galvani, which is his nephew, Giovanni Aldini. And he is probably the reason that Mary Shelley heard about these electrical experiments to inform her writing Frankenstein, because he did the most gruesome experiments trying to keep this idea alive. But instead of keeping it in legitimate discourse, he essentially turned it into like the Daily Mail of the 1800s.
Really?
Oh, it was a disaster. I mean, he did things like he electrified prisoners. These were basically like stunts. These were like TikTok stunts, to go back to our social media metaphor. He got one guy who had just been freshly hanged, and he stuck electrodes in his ears, I think, and one into his anus, possibly. I hate to say, and then it was just sort of, let's see what happens when we do this. And he raised his hand, sort of appeared to point at some of the people, and so he would have these audiences of sort of rich dilettantes who were looking for entertainment, and they would sort of crowd around while he did them. And so it's just gruesome stuff, things that desecrating bodies, and they didn't care because they were prisoners, and then doing terrible things to animals. He was part of a collective that tried to make a battery out of brain tissue, muscle, and hat material. And then that underpinned this huge rise in electroquackery in the early mid 1800s, where, you just had all the chancers coming out. It's almost like the crypto scams of the 1800s.
Sure,
had electric belts to, zap you, which would help you either with infertility or with too much fertility. It went so far that then there was a huge backlash against all of this. And then from there on, it became embarrassing to talk about it, except in the context of neuroscience, because one particular researcher saved the study of bioelectricity by confining it into like a really tight scientific silo of neuroscience. And that was Émile Dubois-Reymond. And he almost blinded himself trying to figure out how to make these, frogs' particular nerves jump in a particular way. Because, he was getting very close to them. He was so obsessed. He was chewing their muscles apart so that he wouldn't risk adding extra electricity from a metal. He was obsessed. But his obsession paid off, because he then essentially guided bioelectricity into a legitimate form of, studying it in neurobiology. And so people did it observationally. But to your question, the reason that the invention of the patch clamp in the 1970s was so crucial is that before Nair and Sackman invented it, we had only observational studies. All we could do was expose nerve here, zap it here, measure some stuff, hope for the best. And so as a consequence of that, we didn't have sort of direct evidence that the stuff was actually electric, right? There were still wars in like the 20s and 30s and 40s. There was this whole thing called the soups versus the sparks, which sounds like the worst version of the West Side Story, but they were sort of, the people who were like, no, everything in the nerve impulse is electric versus no, everything in the nerve impulse is chemical. And they were still having these arguments until in the 1970s, two researchers invented a method of essentially observing the electric signatures of ions moving in and out of cellular membranes, which finally put to rest the question of how an electric impulse passes down a nerve cell.
And then eventually not just a nerve cell, looking at every type of cell.
Yes, but that took a little bit longer.
That took a little bit longer. But and just so people appreciate, I mean, there is a long-standing sort of, I don't know if you call it a problem, I suppose, where you touch an oven, you move your hand away, and in such an incredible speed, that sort of thing could not be explained by chemistry alone. There has to be a sort of quicker way to communicate the information, which ultimately ends up being electrical impulses coming down a nerve and through muscles and that sort of thing.
Right. And so then, those are the ways that the exchange of types of ions in cells occasions a sort of very powerful voltage switch in a nerve cell, which is how the impulse gets passed so quickly from your hand, up the nervous system into your brain, to then occasion a really swift reaction as well. So that does account for the speed of the nervous impulse.
So we get to a point with the patch clamp.
Sorry, I just want to make sure. Sorry, please. I just want to make sure it's both. Like, that's the thing. It's both.
Of course,
it's electrical and chemical. Yes. I just want to make sure I'm not. Yeah. Sorry. Go ahead.
Yeah. No, we get to the point with the patch clamp where we now really sort of understand it and put that debate, that schism to rest. And now, and this is a very interesting concept that you lay out in your book, is the idea of electrical identity of cells, right? This is something that most people, I would venture to guess, even scientists who, like myself, doing research, sitting in front of tissue culture, you're in your silo, you're thinking about maybe genetics, you're thinking about the cell cycle, you're thinking about everything but electricity. And you're looking at these cells and you realize, after reading your book, I realize that, every type of cell has a sort of different electrical identity. You have mature cells, you have cells that have not proliferated yet or are proliferating, and then you have cancer cells. And there is a big disparity between the sort of electrical currents that run in and out of these cells. Can you tell us a little bit about that? Because I think it sort of sets the stage for this idea of a unifying theory, which I find most fascinating coming out of your book. So if you could tell us a little bit about that, that would be great.
Well, right. So we'll start with a tiny little backstory on ion channels, because I think that's important. So back to the nerve cell. Now, a nerve cell, it's sort of resting potential, which I like to think of as like its happy place, where it doesn't need to change anything. It's just, that's where it lives. It's at minus 70 millivolts approximately, give or take, with respect to the extracellular electrical identity of the fluid out there. The reason that this happens is because nerve cells, like all cells, they are encased in a membrane, and the membranes are studded with tens of thousands of these little pores called ion channels. And these are like little bouncers at a club. They're smart. They know who's on the list and who isn't. And normally they like potassium and they don't like sodium. And so the sodium ions, they sort of keep out much more than they keep out the potassium ions. And then that their selectivity creates a specific type of commingling of ions inside the cell that creates a voltage at the membrane, a voltage difference of that minus 70. And that's a nerve cell. And the action potential is when all the exit doors open, or the emergency doors open, and it flips to zero, and sodium goes in and potassium floods out, and then they try to renormalize. Quiescent cells that are not actively zapping also have membranes, and they also have ion channels. They're not excitable, not quiescent, sorry, they're non-excitable cells. They also have ion channels, but they have different preferences for different kinds of ions. And as a consequence, they tend to have slightly different potentials. I don't have my sheet here, but I know that rubric
we'll put it up on the screen. We'll put a figure up on the screen.
Okay, so great. Yeah, I'll send that to you. So the rough rubric is, the rule of thumb is that cells that proliferate have a much higher absolute value of voltage potential. So you've got bone cells being about, I think, musculoskeletal tissue is about -90. Then you've got things like skin cells, also a little bit around the sort of 65. You've got nerve cells, -70. And then as you get into, and these are all sort of, they've established their careers in the society of cells, that is your body. Nerves aren't going to turn into something else. Then you get a little bit lower, you get your fat cells, which are a little bit hovering a bit more close to like -50. Liver cells are about -35, -40, which is super interesting because liver cells are much more regenerative than nerve cells. And then all the way at the bottom of the scale, you get proliferating cells like stem cells and newly fertilized embryonic cells, and eggs, I think just sort of egg cells. So you get things that are very, things that have a lot of more potential, they do not have a high resting potential, weirdly enough.
Right.
And, but so, which is very sort of, it just puts me in mind of a lot of the things that we tell our kids, like you have so much potential,
Yeah.
And then as they sort of achieve these little, what they do in the society of cells, their identity is crystallized electrically. But then the weird thing that happens is that when those cells, when whatever it is that happens to them to turn them cancerous, they shake off their electrical identity along with that, with like what they're doing in your body. And then they're like, screw this, I'm going back to like a unicellular type of state where all I do is eat and multiply, and I don't care about what happens to the rest of the body. I'm just in it for myself. And they drop immediately down to this infantile, like zero-ish, well, much closer to zero resting potential. And so this was something that they didn't really pursue in an organized way when they first got the patch clamp, because it was so far outside the narrative that existed that bioelectricity was only relevant in the nervous system, which had saved the study of bioelectricity in the 1850s, but then sort of doomed it to this very narrow, this very narrow expression in science in the 1900s. So
and also at the time of the patch clamp, in the early time of the patch clamp, I don't think, correct me if I'm wrong, they had any sort of means to manipulate these ion channels at will the way we do now.
Definitely not.
So it would have been difficult to study it thoroughly the way we can study it now.
No, absolutely.
And a great example of that, you're talking about cancer cells flipping, is Brooke Charnay in her a study of him. I'm sorry,
no, okay.
Of course. Being able to revert cancerous cells by manipulating in tadpoles their electrics.
Yeah.
And so I know it's still a big area of study and a little bit controversial, but it definitely, the data, and I'll put the paper up on screen, is a fascinating read.
It is.
Especially for somebody like myself. I was working in a cancer lab, working with tissue culture and manipulating genetics. But as Dr. Levin, brought to my attention and reading his stuff, a lot of this stuff, most of the stuff happens outside of genetics without any alterations to the genetic code.
So you're not altering the genes, but it is all connected, right? Because I mean,
Yes.
there are again, sorry, I don't have this committed to memory. I was just talking about sort of some of the genes that change when the electrical identities of cells change. This is some work that was done by Min Zhao. I can probably give you that paper. It was from 2006. It was like the big first one when it came to bioelectricity and wound healing, to be like, here's what happens when the body's endogenous electrics get involved in healing a wound. Here's the gene changes that you see. And that was a big breakthrough because, there you suddenly saw how it was connected, because obviously having the genes and what happens to them is that was a huge part of legitimizing bioelectricity research. Because before that, it was really easy to sort of lump it always it got lumped with those people with their sort of electric penis belt zappers or whatever. Right. And so after, people, the sort of slightly unsung heroes there who sort of worked despite people always disbelieving them and always being skeptical and sometimes quite rude about their work, they sort of persisted just regardless of all of this. And they found the genetic mechanisms that were being enlisted. And so that suddenly then started tipping people into being like, oh, there's really something going on here.
Right. And let me just correct myself when I said no changes on the genetic level. Yes, expression of genes go up and down, but there's no writing of new genetic code.
Right. Exactly.
When we start dealing with really mind-blowing stuff like being able to have a two-headed planarian worm, right?
Yeah.
This is happening outside of the genetic code.
Right.
And something that you mentioned as well is wound healing. I mean, and you lay this out very nicely in your TED Talk. When you have a wound, a cut in your arm, there is a large change in the electric potential at that area, which sort of signals the immune system to bring in the cells that are required for healing. So just a little bit about that, if you don't mind.
Sure. When you cut yourself, so the skin cells, just like nerve cells, they have the same sort of, delicate, happy place balance of ions and their little minus 60 something membrane potential. And they're also, as you know, like epithelium is like locked really tightly. Cells are locked very tightly together by way of these little guys called gap junctions. So I like to think, obviously, this is the barrier against the world. This is our membrane. And when you cut that, you basically are making a short circuit because all of these cells, all the ions leak out along with everything else. And so this creates a current of injury. And this creates a voltage. And the electric field generated is something like around, it can be anything from like 40 to 200 millivolts per millimeter. But this thing, it's like this initially, this was, I think this was all put together by a scientist called Richard Nuzzitelli. He invented this thing called a dermacorder, which was something that could actually measure this, not as previous probes had done, which they needed to be like sort of stuck in a cell. He was able to do this on a sort of dry skin, like just, in a normal environment. Yeah. So what he found is that this initial field, this electric field, its cathode is right at the center of your wound. And it's calling all of these like cells, like keratinocytes, to help rebuild, and like the macrophages that help mop up the mess. And they call them all in. And when you first get this wound, initially, the field is the strongest that it's going to be. But as your wound heals, the electric field gets smaller and smaller and smaller, and until when you're all the way scarred over, it's all gone. And the other thing he found, which I found really intriguing, is that in people over the age of 65, that initial electric field will be about half the strength of what it will be in people under 25, which I think that's so interesting because I think it correlates with a lot of people's experience. As they get older, their wounds heal more slowly.
Right.
And so a lot of people are looking at this for trying to figure out why some wounds don't heal, like non-healing diabetic ulcers. There's people who are looking at trying to figure out like what goes wrong in this process, and can we sort of electrically juice it a little bit. And there've been, it's still early days. There've been some studies that look really good, some studies that look like we're still trying to figure out how to engage it.
And to your point, hopefully most people haven't experienced this, but up until about the age of 11, if you cut the tip of your finger off, that'll regenerate, and then you lose that ability to do that.
Yes.
Right. And so that may have something to do with bioelectricity. Now,
I think it's all part of the continuum because, we start as these like perfect regenerators when you're first developing, when you're just like, two cells, if somebody cut you in half, you become twins, right? It's like a planarian, these masterfully regenerative creatures. So you can kind of map a human according to their analogs in the animal kingdom, some of which regenerate really well, like starfish, and some of which don't at all, like dogs. But you can basically, we're patchwork regenerators across time and space. We start out being like planaria, and we end up later in life being just, the only thing that really regenerates is our intestines, our liver, and our fingernails, basically.
Right. And you mentioned body shape or a sort of development. This is another sort of application of this idea, this unifying theory, is we know that DNA does not contain the spatial information to sort of tell developing embryos that eye should be here, gut should be here. That information is not coded there, it's coded somewhere else. And it might very well be bioelectricity, which now you start to develop the idea of the bioelectrome. Can we sort of, for development, for regeneration, for wound healing, can we sort of map this whole constellation of networks to be able to ultimately read and write it, the sort of way we can read and write software, which is, I think, the end game, the end goal for this whole idea?
I think, many people are working on their own small, or not small, but their own
niche
of this. But I think for Mike Levin, I think that's his sort of, he's pursuing this idea of being able to read and write what he calls the bioelectric code, and the bioelectric code being, because it's, I focus heavily on the electrical identity of cells, but cells aren't the only thing that have electrical identities. Your organs also do, like that skin battery that I was just talking about. That's the organ of the skin. It uses that to repair itself. The heart uses its electrical sort of identity as a collective to pump, blood to synchronize itself. So that's, liver, every organ has its own sort of electrical identity in the ways that it presses bioelectricity into service. But then there's also, we've started seeing it, outside of, humans. In fact, this research actually predates a lot of the research on the human electrome, because for a while, plants were the thing to study when you looked at bioelectricity. Even like in the 1920s and 10s, like Jagadish Chandra Bose was studying the plants. There was a big constellation of big names studying the electrical signatures of things like onions and trees and flowers and algae. And now, I think this new crop now of this new guard of bioelectricity researchers is trying to put together an umbrella to understand how all these things are connected. Like, how is, cellular electricity in humans connected to, the way that bacteria use electric signals to coordinate themselves into biofilms, multicellular collectives. And there's actually been some really interesting research out of the University of Bergen looking at how these little aquatic doodads, I forgot their name, it's something flagellates, but they're considered the closest relatives to to mammals. They use also bioelectricity as a kind of choreographer to take them from single cells to collectives. And that's always the big question. How did single cells decide to team up into these, societies where everybody has their own job and it works really well? And, how does it work if they all have the same DNA? How do they all have different jobs and phenotypes? So.